So I'm Andy Hall. You've had the misfortune of seeing my photo about 20 times, which is a sort of recurrent nightmare. And it's also rather out of date, as you probably noticed. So my background is actually as a haematologist, somebody looking after blood diseases rather than GIST. And I am old enough to remember days before imatinib. And as you probably know, imatinib was actually developed first for patients with chronic myeloid leukemia. And when I was a trainee haematologist, if you had a patient coming in with CML, often in their 30s and 40s, then you knew that their diagnosis was at best sort of maybe 2 or 3 years, and the treatment was going to be quite tough. And then imatinib came along and actually just transformed that from a disease which was almost invariably fatal within a few years to something which was treatable. And at that point, I realized, there's a connection between taking a drug and it working and the biology. So the thing about hematology is you're very close to the science all the time. And there's a tradition that hematologists and science goes quite close together. So I was very intrigued by the mechanisms that worked behind a disease. So I decided I'd do 6 months of research, and I forgot to go back to the clinic. So I'm worried there's a big queue of people. So I say, he'll be here soon, I'm sure. So I got involved in laboratory research looking at children's leukemia, actually, and ways in which that treatment could be improved. And again, it was a science-based thing, so I actually ended up with a white coat in a lab with lots of nice fancy equipment. And throughout that time, I'd always had a close link with the patients because, as you probably know, as you definitely know because of GIST Cancer UK's support for research, a large percentage of research money in the medicine comes from charities and patient support groups. So just a passing thank you for all the money that's gone into research from GIST Cancer UK for primary research. So I had the privilege of meeting patient groups such as yourself. And the thing that struck me about groups such as this is the strong desire to support research because you can realize that there are still things that need to be done. So within GIST, the big thing that needs to be done from a primary science point of view is understanding what Vicky was talking about, which is what's going on in the patients who don't have the sort of GIST that responds to imatinib. There are plenty of questions within the groups of tumors which do have those mutations, improving those drugs, doing things about mutations that break through that science. But that was kind of one of the things that we were interested in. And what I was struck by those groups is the desire of people like yourselves to partake in research. And on the other side of the equation, there's all these people who are interested in science, who are very interested in finding all the samples and things they need to be able to do that research. But the two sort of groups weren't meeting. So what I decided we needed was some sort of fancy dating organization, right? So we've got a very sophisticated dating. So when I left full-time research and very boring management in university a few years ago, I set up something called RareCan. So RareCan is all about dating, okay? So it's about linking patients who are interested in taking part in research with the scientists who want to take part in that research. The important thing to note is that we are not a charity, so we're not fishing out of the same pond when it comes for the money. The pond we're fishing out of is the money which is on the side of the pharmaceutical companies and diagnostic companies. All right? So the model with RareCan is that people sign up and say they're the sort of person who'd be willing to take part in research or have their samples used for research. All right? And then we go out to the pharmaceutical companies, academic groups, and people like that, and say, we've got this group of people and samples who you can use to make improvements in treatment. So that's our model. Okay, where am I in my talk? So this is all about participation in research and people working together. So there's somebody called Leroy Hood who set up this idea of 4P medicine, okay? We always like a list of 4 Ps, 3 that, 5 of this. And so the point that Leroy Hood was making, it's nearly 20 years ago now, is they're all individuals, okay? So we tend to have to lump things together in science, but actually at the end of the day, you lose some of that individuality. Next slide, please. Okay, so what are the 4 Ps? So the first one is predictive. So we all have our own, unless you've got an identical twin, I should have sort of something, unless you've got an identical twin, we've all got our own unique genome, so we're all uniquely different. We know, looking around the room, you're all different. So that's the first P. For the second P is preventative. We should all think about making sure that we do the best to make ourselves as healthy as possible, and we eat as well as we should, we don't smoke, we don't do horrible vice-like things that make our future not so good. Okay, the third is personalized, so that really is like predictive in a sense. Again, the treatment is individual. So the stories we've heard today on the video and from the people who asked their excellent questions were individual stories. Each of you had an individual problem. And why we're so proud of our GP system, when it works well, is that we've got a GP who can take those things into consideration, okay? But the one that I'm talking about today is more about participatory, okay? So this is changing a mindset. Next slide, please. Okay, so in the bad old days, medicine was about telling people what to do. So you went along to your GP, they may have asked you a few questions, but at the end of the day, they said, you've got to do this. Now it's much more of a dialogue, and we've had several questions related to that. I think one of your questions was, how can I get my GP up to pace about this disease? How can I work with them to make my treatment better? And it's the same with research as well. So you used to talk about doing research on people. Now that's all changed. It's all to do with people.
Can I just interrupt?
You can, my dear.
Several years ago, I was invited to a conference, and I thought initially it was to represent GIST at this conference, and it was in Strasbourg, and the cheapest way to get there was on the train. It was a nightmare journey. I hated it. But when I got there, I then found I was actually within a group of patient representatives where actually I was representing cancer as a whole, and the others were other diseases like diabetes. And then the whole purpose of this conference, then it materialized, was to try and give researchers back their mojo. They were stuck in laboratories, they never really had any contact with patients, and many of them were doing research and actually had kind of lost the plot. Why are we doing it? What's it all about? They had no real emotional connection with it, didn't understand. So this divide that Andy so eloquently describes is definitely there, and obviously what he's describing now is a way for actually us to fill the gap and make research happen faster.
Thanks, Jane. Thanks. That's very true, and actually it's a great stimulus for people. You're right that sometimes scientists do get too remote from what they're doing. I'm being fortunate because I think I've worked in labs where there've been close contacts, and actually it just stimulates you. So I mean, science can be very boring. Okay, you can do lots and lots of experiments and they don't work. And you can get very disheartened, etc., etc. Go and talk to a few patients, and you can come back really sort of G'd up again. So that partnership, that encouragement, just as you talked about working with your GP, work with your research scientist if you can find one. Next slide, please. Okay. So how does this relate to providing better treatments? I think I've alluded to the RareCam model. So next slide, please. And the next slide, please. Okay, so where we fit in is as I said before, it's like a fancy dating agency. And the problem is that patients don't understand that they should actually be very close to a major treatment centre. It's very irresponsible for them to live, 50 miles away from the nearest centre. They should have known better before they chose that particular disease. And so they tend not to be where they should be, okay? And so if you're doing a clinical trial, the traditional way for a company to do a clinical trial is to set up a set of centres around the country and wait for the patients to arrive. Now that works okay for common cancers, so sadly within most forms of breast cancer, a big hospital have plenty of patients. But for rare diseases like GIST, they can wait a long time. And we've spoken to drug companies who set up 4 centres in the UK and not recruited a single patient for a rare disease such as GIST. Now, setting up a clinical trial is horrendously expensive. And so this is a very, very inefficient way of doing it. So what we're doing is turning things around. Next slide, please. Oh, and that's okay. Next slide. So it's about to do with what I call screening and recruiting. So instead, the usual way is to identify a target within a cancer. Okay. So in your cases, it will be the c-KIT gene mutated, and in your lab you found that target. Okay. Oh, I'll make a nice fancy drug to it. I made a fancy drug to it, and then further down the line, oh, I better find some patients to be able to try it out on. Okay, and that's the model that doesn't work very efficiently. Next one, please. You can turn that around by finding your patients first, okay, working with them to assemble enough samples to identify new targets, and then say, I've got these patients, I know where they are, okay, and I know what they've got. You can set up your center in the best place for them to be able to come to me to do the research project. And believe it or not, that's not a model that's occurred to drug companies before. And it's a way that they're now beginning to think that they can make their process much more efficient. Next slide, please. So our model is to obtain that consent, diagnosis, to obtain samples, and to reapproach people if drugs become available. So what we do is we're getting people to sign up to our website. We'll send them a consent form. It's NHS approved bioresource, and then we'll screen them with a genetic panel. So the genetic panel for their tumor shows those common mutations which are now associated with the newer drugs that are coming through. So this is outside of GIST, I must say, because you're an unusual example. And then when a drug becomes available to those targets, we say we've got patients situated here, 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 here. We can contact them back again and say, are you interested in this particular trial? And then we can put the two together, and the dating leads to a happy outcome. Okay. So the advantage from the drug company's point of view, these trial-ready groups of patients, is a big reduction in cost and a big increase in speed. And those are the two things that cost them a lot of money from a financial point of view. From a patient's point of view, of course, there's the opportunity to take part in those trials, and there's good evidence that taking part in a clinical trial is a good thing to do, okay? So if you look at the data of people who are on trials, they always do better than people who aren't on trials. And that's because they go and see the specialist quite regularly, they get a lot of extra care. So if you get a chance to take part in a trial, consider it carefully, okay? So, and also the big point of view, I think, is that greater patient involvement. So drug companies get a mixed feeling in people's minds, I think. I'd be interested to see how you feel about this. We hate them in some ways because they make so much money out of our diseases, and that's terrible. But we want to get treatments from them to be able to live longer and have our diseases treated better. So we have this sort of mixed feeling. But actually, at the end of the day, they're the ones that do develop these new drugs. So they want to open those channels. They want to work with patients to deliver trials in a more efficient way. Okay, next. So this summarizes our model. Hopefully you've understood basically what we're doing. So we've got a fancy website, fundamentally, which you can visit and you can sign up to. All you're signing up to is for us to contact you and keep you informed about what's going on in your area. It covers all rare cancers, and there are nearly 200 flavors of rare cancer, okay? These are cancers which occur at less than 6 in 100,000. So there's a lot of rare cancer around, actually. It's about 20% of the total of all cancers. So individually they may be rare, but as a group, they're not. So your family, as it were, of people with unusual tumors is wider than you thought it was, because there are other people with similar sorts of issues in terms of isolation and feeling that there's not much interest in your area. And then we bring those partners together and we talk to our commercial partners. Next, please. Okay. So we've been doing a pilot project to see whether we can actually make this attractive sounding proposition work in practice when it comes to getting samples. So as I said before, samples are really, really important for driving research, okay? And all of you who have had a diagnosis of GIST, all of you without exception, I would hope, would have had a biopsy at some stage or a tumor looked at under the microscope, because you can't actually diagnose these things without doing that. And a bit of that material, I'm sure you're aware, will actually be kept in the pathology department. So there's a bit of your tumour probably sitting somewhere in a little bit of paraffin. And they keep those tumour samples forever, actually. I mean, it's at least 30 years. And those tumour samples are actually very valuable. They keep them because they may need to go back to them to look at them in the future for your clinical management. So they'll always hang on to a bit, but often there's a lot left over. And that leftover bit can be very useful for research, but you have to give your permission for that to occur. So the NHS will not pass it out to anybody without your written consent. So we contact people and have said, would you give your consent for a piece of research? And we were interested to see whether we could get samples sent from pathology departments to our lab in Newcastle we're working with, to have it processed, to have some of the tumours scraped off, and then put through something called whole genome sequencing. What that does is look at all the genes, which are the bits of the DNA that code the proteins, and the proteins are the bits of the cell that make you work and make you sit there and wish it was about lunchtime, because those are the proteins that are active. We need to look at those genes to see if they're normal or not. And a way of doing that is by a laboratory system called whole genome sequencing. And we had a group in Teesside led by an ex-colleague of mine called Professor Vicky Rand, who was prepared to do these samples for us. We thought, what samples should we look at? And I heard a little voice in my ear with a Bristolian accent. So, you know, it might be yours. So we decided we'd concentrate a lot on the GIST, and in particular the wild-type pause GISTs, to see if we could find any novel mutations that are driving the process. So some of you in the room I know have very kindly given your consent for that to happen, and we managed to get samples sent for that process. Next slide, please. And that was all due to this powerhouse of advocacy, I would call it, the Pause GIST group, who have been had the privilege of knowing and working with for several years now. So thanks very much to them. So we sent out invitations to the people signed up with us, we call members, and said, we've got this project, are you interested in the sequencing project? If so, please get in touch. And then if they said they were interested, we sent them another set of documentation to read, which would be approved by the Research Ethics Committee of the NHS, for them to understand exactly what we were proposing. Okay? And then if they signed that, we then went about the task of getting the pathology sample dug out of the pathology departments, and sometimes these samples are actually stored off-site. In fact, there are disused mines in Wales that are used for storing these things. They are piling up. There's quite a big task storing them. So they've been sending them to us. Then they take thin sections of that paraffin block, look under the microscope, take a scalpel, scrape off the bit which was a tumour, dissolve it, extract the DNA, and send it off to my colleague in Teesside. Okay, next slide. So I'm quite interested in, and that's where your participation is required. No luncheon will be provided until I have feedback from all of you individually. And we sent out just over 100 invitations, not entirely to GISTs, with lots of other sort of tumour types as well. And I think we got 28 or 29 people who signed up and said they were happy to have it. And we are now going through the process of getting the NHS to send the samples through. And it's been less of a problem than I thought it might be, to be honest. So we've had not a lot of resistance. We've had some slowness, and sometimes it's difficult to know whether that's going to be slowness that is going to persist after COVID. So pathology departments, like every other department in hospitals, have been under a lot of stress, and this is not treatment patient care straight away. So we can't jump up and down and say, you've got to do my project, you've got to do my project. And we've had 10 samples sent through for a start, and there's some others with colleagues in Cambridge I know we're going to enrich. And those samples are now being processed. So I haven't got any results for you today on that, apart from the results of this interesting process. So, your luncheon task, what is the time, Jane? Oh, we're okay. I wouldn't want to, yeah, okay. So I've got 3 sort of separate sets of questions, and it would be quite nice if you sort of went into your table and had a little sort of chat, It's not an exam question. There's no right or wrong answer here. I'm just interested. So what would your barriers be to signing up to RareCan? So one of the barriers you may have is a company, it's a commercial. This chap with a grey beard is going to make money out of my samples and all this sort of thing. That's terrible, it's awful. His shareholders, etc. It's not the sort of thing that it should be done within the NHS. It should be done with a charity, which is fine. But interested to know if there are other barriers, maybe not quite understanding, maybe you're worried about the data not being secure, that sort of thing. So it'd be really useful to get that feedback, how strong that is. And please don't be polite. Don't be too rude either. I don't need to go over the top, but just tell me. What would you stop giving consent for samples? So okay, if you were happy with that, what's your worry about a sample being used? I mean, is it that it might not be there for your future treatment or something like that, or is something else, or don't quite understand, or losing control, or something like that? And then this is the more tricky one. This is genuinely a difficult ethical debate. Would you want to see the results of your sequencing? Okay, so if you're going to spend time on this last one. Now, the MRC currently recommend that you do not pass. The Medical Research Council of this company. They recommend that you don't actually send the results back to patients because you're uncertain what those results mean. They are not done in a clinically graded sort of way, so they might be unreliable. And that would cause unnecessary anxiety. But I would want to see mine. So I'm interested to see what your feeling about that is, and how we should do this. There's no right or wrong answer about that either. That there's a tricky thing that if you found something in the genetics which had an implication for your family, then that has a knock-on effect on somebody else. And how you deal with that is the whole business of genetic counselling, which is quite tricky. So it's one of those genuine ethical dilemmas. So if you have your energy is directed to the last one, be fine. So if you could sort of gather around your tables, and Jane and I are going to circulate around to make sure that you're actually talking about this and not something else. And anybody caught talking about...
Point of view, we can have one person